All right, so we're going to continue the notes that we started last week. We left off after the dihybrid cross, which we'll end up going over again tomorrow. But right now we're going to talk a little bit about probability. If you had stats, then you've had this before, which a lot of you guys had. If an event is definitely going to happen, then they say the probability is equal to 1. If some type of event cannot happen at all, then the probability is going to equal 0. So, any other probability, all other events, are obviously going to have a probability between 0 and 1. Okay, so suppose you have 2 coins, a dime and a penny, and each flipping those are separate events. They're independent events. And so when you flip the coin, it's obviously a 50-50 chance whether you get the tail or whether you get the head. And so that's what they wrote right here. The chance of getting the head, the probability is 1/2, and the probability of getting the tail is 1/2. And the same thing for the penny, right? It's 1/2 for each. All right, so this is just like our monohybrid cross. The chance of getting both heads, right, is 1/2 times 1/2. That's called the multiplication rule. So in order to get both of them to be heads, you would take those probabilities and multiply them, and 1/2 times 1/2 is 1/4. Same thing for getting both tails. They're both 1/2 probability each. That's a multiplication rule. You're going to multiply those together. It's going to be 1/4, or 25%. So then you look at, well, what if I get one of each? What if I get one head and one tail? Where there's 2 ways to get a head and a tail, because you can get a tail dime and a head penny, or you can get the head dime and the tail penny. So in that case, because there's 2 different ways to get it, that's called the addition rule. So that's going to be a quarter plus a quarter equals 1/2. And so that's called the addition rule. Okay, the next topic that we need to go over are pedigrees. Okay, pedigrees, there's a couple symbols that you need to know, and actually they are over here. A female is always going to be a circle, and a male is always going to be a square. And then when you show the marriage, you're going to have a horizontal line going across, and then to show an offspring from a marriage, you would have a vertical line going down. And then if they have more than one child, then you would have like a comb-looking structure to show that they had 2 boys and a girl. And then you would always have your generations numbered by Roman numerals on the left. All right, so there's different kinds of pedigrees. Some pedigrees can show dominant traits. Some pedigrees will show a recessive trait. Some of them will show a sex-linked trait, which we haven't got into yet for our Punnett squares. So this one in particular, shown here, is a pedigree showing a dominant inheritance. So looking at generation 1, you have your parents, where you have the father that he has a dominant trait, and the mother did not. And so they had 4 children. They had 2 girls and 2 boys, and you can see that the girl and the 2 boys got the dominant trait, and the one girl did not, and she ended up being recessive right here. And then you can see their grandchildren, the marriages, from the 3 different kids had kids. So you can see how the dominant trait is showing up in every generation. And another little key, or hint I should say, is that it says half of the offspring usually of an affected parent are going to be affected. So if you have a dominant parent, then half the kids usually are going to be dominant as well. And if you do a Punnett square, you will see how it's a 50% chance of getting it. And so what you're going to end up seeing in AP Biology is that they're going to give you a pedigree, and then they're going to ask you, is this pedigree showing a dominant trait, a recessive trait, is it sex-linked, and so on. And so these are hints so that you can figure these things out. Okay, so here is a pedigree that is a recessive one. All right, so let's say in generation 1 we have a heterozygote. So you can see it's half shaded in. Heterozygote means like big A, little a. And so you can see that the heterozygote trait will get passed. Like if you're heterozygote, your offspring can be heterozygote. And you can see how that got passed down to the kids and then the grandkids. And then you can see here we have a recessive individual. So to recognize recessive, it's not going to show up in every generation. It also says that only 1/4 of the children in a family will be affected. And if you did the Punnett square, you could see that it would be a 25% chance of getting the recessive trait. And also you can see that affected people who get the recessive are going to have parents that aren't affected. I mean, they're heterozygotes, but they do not have that trait. So those are the hints to figure out a recessive pedigree. Okay, this is just a little introduction to a mutation. We're going to get into the different types of mutations later on. But mutations are very rare. They happen in 1 in a billion, usually. But they're inherited. They can be inherited. Changes, they're errors in the DNA, changes in the genetic material. They occur on genes, and so depending on what type of gene it is, will determine what type of mutation and what the effects are for that. So one letter or allele, like the little a, can be mutated and changed to a big A, for example. And so doing that obviously is going to affect that person and that gene. Okay, so we're going to get into one of the specialty problems for genetic problems, and it is called incomplete dominance. Okay, and so this is when you have two traits that both of them are not dominant. One is not dominant over the other. So what happens is they blend. And so they call that an intermediate phenotype or a blend. Okay? And one major example that they use in biology is the Japanese 4 o'clock plant, which is also called the snapdragon. And so these genotypes right here, you need to memorize and you need to know. You need to be able to apply that type of genotype to a incomplete dominant problem. And so for a Japanese 4 o'clock plant, these are plants that have red flowers. They can have white flowers. But then if they get one of each gene, it will blend. Because red is not dominant over white, and white is not dominant over red. And so to be red, you would get a big R, big R. There are no little letters here. To be white, it would be big W, big W. And so if you get one of each gene, which is a big R, big W, then that's how you get a pink flowered snapdragon. So for example, let's cross a red flowered Japanese 4 o'clock plant. So red would be represented by big R, big R. And you're crossing that with a white flowered 4 o'clock plant. And so that would be big W, big W. So you are not allowed to use any lowercase letters on an incomplete dominant problem. All right, so you bring the letters down the same way that you would any other monohybrid problem. And typically for this type of problem, you would always write the R first. And I don't know if that's an alphabetical thing, but that's just how I've always seen it. So you bring the letters down just like you would before. And then obviously they'd ask you some type of problem. And we will see these tomorrow when we start doing our genetic problem packet. So you can see that all of their offspring are pink, which is our W. Even though the parents were red and white, all their offspring are going to show up to be pink. Okay, so this is an example of an incomplete dominant genetic problem. So here's an example of it all done. And you can see if it asked you for a genotype percent for the offspring, you would look inside these boxes, and then you would write 4 out of 4 is RW, or I like percents, 100% RW. And then if it asks you for phenotype, that's what the plants look like, and that's when you would say 100% pink. So you will see these and do these in the next day. Okay, we're going to get into specialty problem number 2, which is called codominance. Codominance is when you have two alleles that are going to share dominance. So they're both dominant. One is not dominant over the other, They're both dominant, but you also have a recessive going on as well. And so this happens in blood types, blood typing for humans. So a little bit of background. In humans, there's a gene, and they call it I, gene I. Gene I will, and we haven't learned this completely yet, will encode a protein. It's actually an enzyme. And that enzyme is responsible for attaching a sugar to the surface of the red blood cell on the glycoprotein. So it's like attaching a carbohydrate to the glycoprotein and turning it into a, well, it's already a glycoprotein, so it's adding more sugars to it. And so there's a gene called I that does this. There's actually 3 different alleles on that gene. Okay, so on gene I, there are 3 alleles possible. It can be IA, and usually that is written as a superscript. So I, like to the A power, so it's not written on the screen, right? I to the B power, or it can be IO. Okay? All right, so what happens in codominance is that IA and IB, those alleles are actually codominant. But what they do is they will encode active enzymes, but these enzymes attach different sugars to the glycoprotein. Okay, so there's different sugars involved for the A and the B alleles. And then you have our third allele, which is the IO. The IO allele doesn't encode an active enzyme at all. There's no sugar that's going to be attached for that allele. So, when there's 2 different alleles present, both those alleles will be expressed. Another thing to keep in mind is that the A and the B glycoproteins are antigenic. And so that means that the recipient is going to mount an immune response and produce antibodies against the opposite letter. So that means that A is going to produce antibodies against B, and B is going to produce antibodies against A. So the blood will attack the other type of blood. This is what we're getting into. The O glycoprotein does not provoke any immune responses at all. And so that makes people that have this blood type O, which is called IOIO, good blood donors. Okay, on the bottom of your outline that says blood types codominance, you have some space there, and you will need to draw this chart in. So you're going to make a data table that has 5 columns, and about 4 rows. Okay, so I'll draw that in. And we're going to distinguish between these 4 different glycoproteins, or technically the types of blood in a human. All right, there are 4 blood types possible in a human. So your last 4 columns are going to have those blood types. So up at the top, you're going to put blood type A, blood type B, blood type AB, and then blood type O. And then your rows going across are going to show what the red blood cell looks like, what antibodies are present in that plasma, which is a part of the blood, and what antigens are present. Okay? So I'll give you a minute to set up your data table. Make sure you have room to draw. So a decent size square. You should have enough room on the bottom of that chart, or on the bottom of that outline. All right, so we're going to start with blood type A and work our way down so you can understand this. For blood type A, the blood has glycoproteins on it, okay, on the surface of the red blood cell. So you're going to draw a circle. You're going to put the letter A in the middle. And then you don't have to draw as many as they have, but you're going to draw at least one little spike coming off of it. And these are the glycoproteins. And you're going to draw a little circle to represent blood type A. Okay? Now, blood type A is going to attack blood type B. So there are antibodies. Antibodies is a part of our immune system. They attack foreign things in the body. So the antibodies that are present in blood type A look like that picture right here. So you're going to draw a little stick, and you can just draw like a V, like a Y almost, kind of a Y, I guess. You don't have to draw 5 of them in my picture. And so that means that blood type A is going to attack blood type B. And then the antigens that are present in the red blood cell is just going to be that little spike with the circle. And those are called A antigens. And that's present in a blood type A blood cell. All right, moving on to blood type B. For blood type B, I want you to draw a circle and put the letter B in the middle. And then on the surface of that blood cell, you're going to put a little spike, and then you're going to draw a diamond, tilted square looking structure. And so that's what blood type B looks like. You don't have to draw that many of them, you can just draw one. All right, for the antibodies, blood type B attacks blood type A. So the antibodies that are in blood type B will fit a circle in them. So you're going to have like a U-shaped spike. So this little spike thing is an antibody, and the circle on blood type A will fit in that, and it can destroy the blood cell. Same thing with B. This diamond square looking thing fits into there on blood type A, so blood type A can kill blood type B. All right, blood type B antigens is going to be the square or tilted square diamond looking antigens at the bottom. Okay, remember, blood type A and blood type B are codominant. They are both dominant. So if you get 2 of those alleles, then you are blood type AB. So that means you are a mix of blood type A and blood type B. So draw a circle. That's your red blood cell. Put the letters AB in the middle. And on the surface of those red blood cells, you're going to draw both spikes, the circles and the tilted squares, because it contains both. Because it contains both, it does not have any antibodies, obviously. It's not going to attack A blood, it's not going to attack B blood. So you just write none. There are no antibodies in blood type AB. And the antigens are both. They have both the circles and the diamonds. All right, last but not least is blood type O. For blood type O, there are no antigens present. So on your circle, draw your circle red blood cell, put the letter O in the middle, and you can just leave it blank. There are no antigens present. I mean, I know my picture shows the little spikes, but there's nothing attached to them. So you can draw the spikes if you want, but they're empty. However, blood type O contains antibodies. They have both antibodies in it. They have antibodies that will attack blood type A, and they have antibodies that will attack blood type B. Okay? I'll give you another, let's say 30 seconds, and then we will move on. All right, so you might want to squeeze this chart in or take a picture of it from the screen with your camera. But this chart is going to show you what type of bloods a person can accept and what type of bloods it can be donated to. So for blood type A, they have A antigens, they have B antibodies, and you have this chart, we just kind of drew it, so it's the same kind of thing, but it just kind of explains it a little bit more. Blood type A can receive from blood type O and blood type A. If you notice, blood type O, all blood types can receive blood type O. Blood type A can only donate to blood type A and AB. All right, blood type B is the opposite. Blood type B contains B antigens, has antibodies for A. It can receive from O, and it can receive from B. It can donate to B, and it can donate to AB. Blood type AB has A and B antigens. It has no antibodies, so it can receive blood from anybody because it doesn't have any antibodies to attack it, but it can only donate to AB. Blood type O has no antigens. It has A and B antibodies. It can only receive from O because it has those A and B antibodies that will attack other blood types. But it can donate to everybody because it doesn't contain any antigens. So you really have to think about what's going on with those antigens and the antibodies to figure all this stuff out. Okay, you have this chart. This chart also is kind of saying the same kind of thing. It's showing what happens during the reaction with the antibodies. It also gives you the genotypes possible. So for blood type A, they can be homozygous blood type A, which would be IAIA, or heterozygous blood type A, which would be IAIO. Blood type B can be homozygous blood type B, which is IBIB, or heterozygous blood type B, which is IBIO. And then blood type AB, there's only one genotype that you'll use for the genetic problems, that's IAIB. And then blood type O is written as IOIO. Okay, so now we're going to attempt a genetic problem. Nope, I lied. We're going to do the RH factor first, which explains the pluses and the minuses in blood types, and then we will do the genetic problem. You do not have this on your outline. I can get these slides printed off for you, if that will help. So you can just listen for now. All right, so there's something else on the surface of the red blood cells. There's another antigen present, and they're called RH factors. If you have this extra antigen, which is a protein, on the surface of your red blood cells, if you have it, then you are considered to be RH positive. And obviously, if you don't have it, then you are RH minus. What does this matter? Well, it matters for pregnant women. If a pregnant woman is RH minus, is carrying a RH positive child, that can be very dangerous for the pregnant mother. Because during delivery, natural childbirth, and so on, the blood is going to mix. The mom's and the child's blood mixes. So what happens, and when that mixes, is the mom's blood is going to produce antibodies for that other blood, because it's foreign, really. And it's fine for the first child. The first child, nothing's going to happen. We're going to be okay. However, now that mom has those antibodies that will attack that positive blood. So if they get pregnant again, and the second child again is RH positive, then you can have some serious complications. The antibodies that the mom has is going to attack that kid's blood. And so what they can do is they'll test for this. They can treat it in early pregnancy. But what happens is the child will not be able to produce red blood cells. And obviously, you know, if you can't produce red blood cells, you don't have the hemoglobin properly working, you're not going to be able to transport oxygen throughout the body, and death could result. Okay, so this is just showing the procedure, what happens. The dad is where you're getting it from. So because the mom is negative, the dad was positive, so the baby became positive during birth. The mom produced positive antibodies during the delivery. The first baby was okay. So you can see the antibodies got produced right here. Whoops. Right? You got antibodies made. Okay, and so then if she gets pregnant again and they're positive, that blood is going, those antibodies are going to attack it. And her anti-Rh antibodies will cross the placenta and can damage those and destroy those fetal red blood cells, and then the baby won't be able to breathe. Okay, so now, as promised, we can do our genetic problem for blood typing. All right, so let's make a 4-square problem, and we're going to use the genotypes that were shown in our chart earlier. We're going to cross a person that has blood type AB, so that is IA, IB, and we're going to cross that with a person who has blood type O, which is IO, IO. In genetic problems, we're not going to be dealing with the pluses and the negatives in the actual problem. So now, when you bring the letters down, you have to put the dominant letter first. So the A would be written before the O. And you got to write the I because that, represents the protein. IB, IO, IA, IO, IB, IO. All right, and then again, the problem would probably ask you, what are the genotypes of the offspring possible? And you would write 50% IAIO, 50% IBIO, and you would have to answer whatever questions were present in the problem. So tomorrow when we work on genetic problems, we will see these in action. Okay, here's an example. This isn't formatted right, but what I want you to see is the genotype percents written out without the superscripts. And then if you were doing phenotypes, you would just say what kind of blood type they were. Okay, one other concept that we need to go over is called epistasis. Epistasis is kind of cool. This is when you have a phenotype expression of one trait is actually influencing another trait. Okay, and so the best example way to learn this is through looking at dogs and the Labrador retrievers. Okay, so here's the genetics part. You have black and brown labs, right? And so that's represented by the allele big B. Allele big B is going to give a black Labrador retriever, and it's dominant over the little b's, which is going to be a brown or chocolate Labrador retriever. All right, however, you have also a pigment trait in them. It's called pigment deposition in the hair, and that is dominant, and that is represented by the letter E. So allele E, which is pigment deposition in the hair, is dominant to little e, which is no deposition. And if they don't have that pigment deposition, then their hair would be yellow, and that's recessive. So if you have a big E, or a big E, little e dog, which means you have pigment deposition in the hair, and they also have the big B or the little b, big B, little b, they're going to be black. But if they have the little e, does not matter if they have the capital B or not, they will be yellow labs no matter what. That little e kind of takes over and doesn't even matter what your big B allele or little b alleles are. It doesn't have nothing to do with that, because little e kind of dominates the whole thing, and then you'll end up with a yellow lab. All right, so for example, the black lab there, that's a dog that has the allele big B for black, and it has the big E for the pigment deposition. To get a brown lab, you would get the capital E for brown, but you would have little b for brown, and a capital E for the pigment deposition to be present to get that pigment. However, it doesn't matter what alleles you have for B. If you get the little e, then you're automatically not going to have that pigment, and you will turn out to be a yellow lab. And so you can see a cross between, it looks like, two black Labradors, and you can still get yellow puppies and brown puppies in that mix. Okay, the next page is showing you Locust. You want to title that Locust. I think I've shown you this picture before when we did mitosis and meiosis. This is showing two different chromosomes, and it's just showing the position of the alleles on the chromosome. Notice these are homologous chromosomes, one from mom, one from dad, let's say, and you can see the P gene and the A gene and the B gene are located in the same position. Those position of where those genes are located are called loci. Loci is plural, locus is singular. There are some genes that are linked and will be carried and passed down from, let's say, like parent to offspring together. They're called gene linkage. The concept is called gene linkage. And so it's an association between genes on the same chromosome. So they will not randomly assort. They will be connected kind of together. And so the rule is the closer the genes are on the chromosome, the lower the chance that they will cross over and see that linkage happen. So this actual concept, and we'll see this a little bit later in the course, it was studied by Morgan, Thomas Morgan, in the fruit fly, Drosophila. Okay, our last type of specialty genetic problems is the sex linkage traits. And this is the inheritance of a gene that's on the X chromosome. So the female chromosome is carrying these traits. And example ones that you see in biology most of the time is hemophilia, color blindness, and baldness. Okay, underneath your pedigree, or to the left there, I want you to write out these genotypes for a sex-linked trait. All right, if you are a female, and we're just going to do it for hemophilia, females have 3 possible genotypes. So remember, these are traits that are carried on the X chromosome. So every X chromosome has to have the letter attached to it. So females can be X big H, X big H, which would be a normal female. Okay? Females can also be X big H, X little h, which is a carrier female, but she's still normal. She does not have the trait, but she can pass on that little h to her offspring. Or, very rare, she can have the disease and be X little h, X little h. And so that would be, she would be a hemophiliac. Okay? All right, and so then for males, males only have 2 genotypes possible. They can be X big HY and be a normal male. Or they can be X little hy and have the disease. They're called a hemophiliac. Okay, so those are the genotypes that you would use in a genetic problem. So let's say the female is normal for the trait. So I'm going to put X big H here, X big H here. And let's say she marries a boy who has hemophilia. X little hy. What are the chances that her kids will have hemophilia? So you would bring the cross down just like before. X big H, X little h. X big H, X little h. X big H, Y. X big H, Y. So if the mom does not have hemophilia and is not a carrier, but the dad had it, notice that all their female daughters, right, their daughters are all going to be carriers no matter what. So if they have a girl, that girl is going to be a hemophiliac carrier. But if they have a boy child, their boys will be fine. Okay, so sex-linked traits are very interesting. If you were doing this type of problem with color blindness, then you would just be switching that H for a C. The genotype chart that you need to know this chart, would be the same format. Same thing with baldness or any other sex-linked trait that you'll see. So again, looking at the pedigree, this is a sex-linked trait. You have females that have the trait but don't express it. So that's what those dots represent in the females. So this pedigree is showing the sex-linked trait, and you can see, oh, look at all those males getting the trait. So there are more males. Males get more sex-linked traits than women do. It's very rare for a woman to get it. So that would be a huge key to figuring out the pedigree, because you would see mostly males on the pedigree with the trait. And so hemophilia, baldness, color blindness, these are all traits that typically men have and not women. Okay, our last page of the notes is just introducing the idea of a plasmid. We have a huge lab with this later on. So refreshing our memory on a prokaryote cell, there's our bacteria cell. Bacteria have one chromosome, as you can see, okay, in the cytoplasm. But they also have these things called plasmids. You can see some of them have one, some of them will have more than one. These plasmids are an extra piece of DNA that bacteria have, but they have special properties. This DNA here could make that bacteria antibiotic resistant. Or it might make the bacteria be able to clean up an oil spill. Or can glow in the dark. So they have like these special genes on them. And so not all bacteria have them, but they're really cool characteristics if the bacteria do have them. And so what's neat is that these plasmids can be transferred from one bacteria cell to another, or inserted into a different organism, and then that organism can express those genes. And so that's kind of leading into what we'll be getting to eventually when we start studying the actual DNA.